Okay, so I'm basically going to continue our discussion from yesterday and get to some more advanced topics. So I forgot to put the list here. There are 3 things I want to get today too. If you remember from yesterday, we quickly talked about sort of this vision where we think genome skimming can now become a viable tool for sample identification and beyond. So you can do even things that you wouldn't be able to do with barcoding. You know, what I didn't talk about much, but in this paper we have touched on are things like cost. How do you do the actual sequencing? You know, those are things that I'm not an expert on, so I'm not going to talk about them, but you can read on them in this paper. So this is where we left off yesterday. We have a query, we have a bunch of references, we want to compute distances and do phylogenetic placement. This pipeline here, over here, we went through most of it. Today, I'm going to talk about 2 things, or say 3 things. One is, you know, when we computed these genomic parameters, if you remember from yesterday, there were a little bit of overestimation of genome length and a little bit of overestimation of the coverage and underestimation of genome length. And we said that has to do with repeats in the genome. We are going to talk about the new method we haven't ever published yet. It's on BioRxiv. It's going to be published one of these days. And that method, what it does is it kind of gets at the repeated structure of the genome. So this, what we call genomic parameters, is now becoming a little bit more sophisticated. It's not just length and coverage, but also repeated structure. So that is my first topic. That's my second topic. The first topic I'll start with is if this bag of reeds that we have is a mixture of multiple species, what do we do with that? How do we deal with mixtures? And that's, I'm sure, something a lot of you are, and that's kind of like the first question I'm always asked. And we have some solutions. I wouldn't claim the solutions for mixtures are perfect yet, but we have some stuff, and I'll talk about that. And then one kind of a specific type of mixtures are contaminants, where you have you know, species that are completely different. So when I talk about mixture, I'm talking about like real mixtures of things that you're interested in, but there are also contamination from things you're not interested in, and how do we deal with that? So these are the 3 things I'm going to talk about. Okay, so before I start, any questions from yesterday? Anything anyone wants to ask before I get going? Get the chat box up. Oh, there you go. Okay, so I don't see anything. Yes, about yesterday. So I'm guessing any option of producing support values for tree. That's a great question. We are working on that. We have a solution for getting support values. We literally just like yesterday started writing the paper. So in like a couple of months, hopefully we'll put that on. Like we'll publish that earlier. We'll put it on archive. We do have a solution for support values for the tree. We just haven't managed to kind of publish it. It's just very, very new thing. Thanks for that question. Okay, so mixed samples. So basically the scenario is your query, so your references are not mixed. Your references are still pure genome skims, but you have a mixed sample of several species of interest. Again, not contamination, not things that are completely different, but let's say you have 2 different insects or 3 different insects, and you are interested in identifying insects. How do you figure out what you have? That's the question. So I think we have to think about this kind of with different scenarios. We can't talk about all the mixtures in the same breath. One scenario, very like high level, one scenario is when you have a mixture of very different species. And now when I say very different, I mean something like distance about like 20% or more, or maybe even 10% or more. Somewhere around there. So think about, well, it's different for different organisms, but not members of the same genus, for example. Those are usually less than 20, like almost always they're less than 20% divergence. Now, whether like a family or order is 20%, that's really dependent on your organism. But if you get, for example, if you look at Drosophila, they're about 15%. Yeast, we saw yesterday, they were about 17% from end to end. Very different. Let's say you have, I don't know, a butterfly and you have a bee, right? Those would be more than 20% different. Telling apart reads that are coming from such divergent groups, that's actually a easier problem. And there are many tools that that are probably sufficient. This is not something we have yet tested in detail, but I'm guessing that most of like these sort of taxonomic classification tools can distinguish things that are that far apart easily. So if you have a bunch of reads, if you have a bunch of genomes from, let's say, butterflies and bees, you can create reference libraries for butterflies, and for bees, and you can kind of take each read and see which group it belongs to best. And read classification methods, I think, will work just fine in cases like that. I'm not talking about read alignment tools like Bowtie or Minimap, but more like classification tools, things like Kraken or another tool called Consult, which I'll talk about later today when I talk about contamination. So this problem, again, we haven't tested, but I think it's easy enough to distinguish large groups. When we get to closely related species, so you have two different Drosophila mixed, right? Or two different bees mixed. That's a more difficult question. Let's say within the same genus, for example. And there, I think a lot of existing, like so again, it depends on what you have as reference. If the constituents of your mixture, so the species that you have in your mixture, you also have a reference genome for them, again, the problem becomes relatively easy and manageable. Why? Because you can just take your reads and map them to those genomes, just with whatever read mapping tool that you like, and that, like, whichever of the genomes it matches to best, that would be your constituent. So you can kind of work around it. Now there are complexities here, of course, but conceptually, at least, it's easy. It becomes really difficult even to think about mixtures when you have a mixture of two constituents that you haven't even seen in your reference library. So you have a mixture of two Drosophilas, neither of which is in your reference library. What do you do then? What can you do then, even conceptually? So set aside all the nitty-gritty problems. Conceptually, what can we do? So so let's do some tests first. So we have, again, a mixture of two Drosophilas. We get a bag of reads where reads are coming from one or the other. Let's assume first that we just ignore the fact that this is a mixture and go ahead and use a skimmer, the tool we looked at yesterday, and compute the distance from this mixture to all of our reference species. What would happen then? So like let's also say I know my mixture is of only two species, not more, right? So I'm just like simplifying this stuff here. What can I do? Well, maybe I can simply assume that the closest two match, like the closest two numbers here. So the smallest two numbers here, R2 and R3, are my constituents. Is that a good assumption or not? We don't know. Otherwise, maybe we can just place it with apples and see what happens, right? Maybe somehow from the apple results we can tell. So the phylogenetic placement. So let's test that. We did a simulation where we are basically taking these various pairs of Drosophilas, mixing them together, computing distances to all the all the Drosophila and using one of these two approaches, top two or just apples. Okay, so with top two, so the first scenario we tested is where both of the constituents are actually present in your reference set. So what is in your mix is in the reference set, the easiest scenario. And what I'm showing here on the y-axis is the amount of error that we have after the placement, right? How many edges away are we from the correct placement on the phylogeny if you just take the top two hits, or if you place with apple and take the first and second best placements. And what you see is that the top two actually works perfectly well. Like there was no case where it made a mistake. So we see primary, secondary. So that's basically saying, is it finding the correct like constituent 1 and constituent 2? The name primary, secondary is arbitrary, right? So we have constituent 1, constituent 2. Is it able to find them correctly? Top two always finds them correctly. Apples, most of the time, finds one of them correctly. It cannot find the other one correctly. So far, it seems like this top two approach works. You just compute distances and find the top two. But of course, the interesting case is when one or both of the constituents are missing in the reference case, in the reference data set. You don't have them in your reference. What happens then? Well, the results are interesting. The top two is still able, in most cases, is able to find one of the two constituents correctly, but it cannot find the other one. In this case, the one that is missing, it won't find it correctly, but it finds the one that is in the reference. And in this case, it finds one of them, but not the other, right? If both of them are missing, there is no like pattern to it. It just finds one, but not the other. Apple, similarly, in this case, finds one of them, but not the other. If both are absent, Apple actually doesn't find either one of them that well. So basically, when your constituents are missing from the sample, none of the sort of existing methods, the way like phylogenetic placement methods would work. Okay, so what do we do? Is this solvable? What we want to do is something we call phylogenetic double placement. So I should have had a slide for it. I don't, so I'm going to just try to draw something here. So let's say you have a backbone tree with a bunch of species in it, and you have a query sample. What double placement does is that it takes this sample and it finds two placements on the tree for it. For example, this could be a double placement, and places that one sample in two positions. It's basically saying it's a mixture of these two things. So that's the way we think about mixture identification. Now, this example I draw here is an example where both of the constituents, the blue is a case where both of the constituents are missing. You would get like two placements with long branches. Now, let's say one of them is missing, then you would get something like, let me use a different color. This green thing I'm just drawing where so this is a case where like one of your constituents is missing, right? One of them is very close to something you have, the other is completely like off. And of course, if both of your constituents are in the data set, you would get something like this, what I'm drawing in black, right? So with this framework, this problem, if it can be solved, in principle, it can handle cases where your constituents are not in the database. So conceptually, it's a nice framework. Now the question is, can we solve this problem? Any question on what double placement is? No, okay, so let's move on. Let me clear up this little thing I draw here. Okay, so Okay, moving on. So the way we think about it is the following. So remember, we got these distances from the mixed sample to each of the references. Can we design a magical deconvolver that takes this vector and kind of deconvolves it into two vectors? Basically saying that your constituents, we wouldn't know exactly what they are. So these names, we wouldn't quite know yet, right? So we don't know the names, but at least we can take these mixture, right? Mixture distances and deconvolve it into two different vectors. And these two vectors would correspond to the distances of each of the constituents if you had the constituents as two kind of like separate samples. Can we deconvolve the vector? And that's how we're going to approach the problem kind of mathematically. So to solve it, these kinds of problems are best handled if you kind of reverse engineer them. So we first try to figure out the opposite problem, which is, if you have you have two species, you know their distances to all their references. Can I predict what would be the distance of the mixture to all the references? Can I solve this problem? This is actually an easier problem than the reverse problem. That's why we call it reverse engineering. This is a sort of simple problem to solve. And that problem we were able to solve. So I'm not going to go through the details. And this equation, don't worry about what that equation actually is. The bottom line is the following. If you have two constituent species A and B, and you have a reference R, those three things create like a little phylogeny like this with three distances. And if I can compute what's the distance from the mixture. That's the distance from the mixture right there. I can compute what's the distance from the mixture to each of the to the reference. This is the distance between the mixture of A and B and reference. I can compute that knowing the distance of each of the constituents and the distance of the constituents to themselves. I need to know what's the distance between these two guys as well. Okay, so if I know all of those, I know the mixture distance. So this problem is solved. We know how to solve this problem. Now, once you solve the forward problem, usually solving the backward problem is easy enough. As in, it can be done. And again, so I don't want to go through the details because there's a lot of details, but I want to give you like a lot of algorithmic details. I want to skip those, but I want to give you some of the basic ideas. Once we came up with this model that I just showed you, a very nice theorem followed from that model, which to us wasn't obvious at all. It's intuitive, but it's not obvious. This is a theorem you can kind of mathematically prove, so it's a nice thing. The theorem says that if your reference library includes both of the constituents, under some kind of like reasonable assumptions, the smallest two distances that you are going to get from the mixture to the references are going to correspond to the constituents, right? So if I have a mixture of A and B, and my reference data set does include A and B in it, in addition to a bunch of other things, the distance from mixture to A, the distance from mixture to B are going to be smaller than the distance of the mixture to any other species. That's a very nice, like surprisingly nice theorem we were able to prove. And if you remember our results, the results that showed in the beginning, this actually panned out to be the case in the simulation results, right? When both constituents were present, top two worked fine. Okay, so So that gives us a solution for that case, right? And in fact, not only we know these are the two smallest, we actually know what that distance should be. Like there is a formula for what that distance should be. So we can kind of look for that value. And if we saw it and we see it, we know that we have those, we have the reference in the mixture. Now, what if both one or two of them are missing? So basically, we take that reverse engineered formula and we try to find the best placement that matches that reverse engineered formula. Let me say that in a slightly more precise way and kind of skip all the details. So slightly more precisely, what we do is we take our reference tree that we had and we look at all pairs of possible placements. So right away, this is not like the fastest algorithm in the world. It's nowhere as fast as apples, for example. Okay, so we'll look at all possible placements like this one. Then we try to deconvolve that distance matrix such that the deconvolved values correspond to that reverse engineered formula I showed earlier, but also match the tree as much as possible. And but it won't ever like completely match the tree. The difference between tree distances and deconvolved distances is our error. And so we kind of record that. Okay, so for this like double placement, we had that much error, right? Then we can go to the next double placement and compute its error, right? So we do this for all pairs of placements. And at the end, we take the placement that has the least error, with the error being the difference between the convolved distances and tree distances. I hope that made some sense. If it didn't, that's fine. Just know that we have a way to solve that double placement problem that I described earlier. Okay, so I think that's all I had on the method side. I wanted to show you some more results. Okay, so the tool that we created to solve this problem is called MISA. Like all the other tools, it's on the GitHub. The method came out last year, so you can use it. Before I go to the results, let me see if there are any questions. What is the reference tree made of? The reference tree is made of either genomes or genome skims that you have. So if you have a bunch of genomes and genome skims, so let's say you are studying Drosophila, right? And you have a mixture of various Drosophilas. There are already a bunch of Drosophila genomes, pure Drosophila genomes out there. There are also probably some genome skims that you have. You can create the reference tree of those genomes and genome skims. And we will do that in the tutorial. Won't the error then depend on how many references we have? So it is not dependent on how many references you have. It depends on whether your reference includes the constituents that you have in your mixture or not. If your reference set includes your constituent, the problem is easier. If it doesn't include them, it's more difficult. And of course, I think you are also right that the more references you have, the better. That's definitely the case. But the most important two references to have, if possible, is the ones that are in the constituent. Are the constituents of your mixture. Of course, a lot of times we don't, right? But if you do, then that's an easier problem. Hope that answered the questions. Any other questions before I move on to results? Okay, so let's look at results. So this is basically the same test I showed you earlier. Now we add this tool MISA to the picture, right? So let's go through each panel one by one. The first one over here, this is when both of your constituents of the mixture are present in the reference. And you can see that as our theory predicted, top two works perfectly fine, but our method MISA also works perfectly fine. and apples doesn't. So you don't want to use apples when you have mixtures. When you have one constituent present, top two finds one of them is very off for the other one, but our method MISA finds both of them perfectly in almost all cases. There's only one case when we are one edge away. This is like one edge away from the correct placement. Oops. So that's also good. When both of the constituents are absent, so neither one is present, again, we find one of them correctly every single time. And for the second one, we find it most of the time correctly. Sometimes we are one edge away. Sometimes we're one edge away. There is one case when we are 3 edge away. So when you are 3, right? These trees are Drosophila trees. They have, I think, 15, let's say they have 20 species in them. So by the time you are 3 edges away, that's not a good result. But that's just one case. Every other case, we are finding both constituents correctly. Simone is asking a good question. He or she's asking how do you know whether you have the constituents or not to know which algorithms to use? The answer is you don't need to, because look, the MISA, under all conditions, MISA is as good as the other algorithms, right? So here it's as good as top 2, here is better than top 2, here is better than top 2, and apples is just hopeless, right? So this is just like conceptually, I want to show you like what conditions each method works with best. But at the end of the day, you don't have to decide that in advance. Just use MISA. So if you have two, so this is error, right? You want the error to be lower, and so if you just look at these results, the answer is that MISA would be the best method to use. Keep the questions coming. Any other questions? Okay, so these results, while you guys are typing questions, these results were based on actual genome assemblies, not genome skims. We also did an analysis based on genome skims. These are lice genome skims, real genome skims, not simulated skims. Results, the patterns are basically very similar. When you have both constituents, both MISA and top 2 work exactly correctly. When you have one of them in the mix, MISA finds the correct. So this tree is much larger, it's 78 edges. So even like an error of 2 or 3 edges are not so bad here. MISA finds one of them correctly every time. The second one, most of the time it finds it correctly. Sometimes it's like 1, 2, or maybe even 3 edges away. When both are absent, again, it works well in most cases. Some cases it's 1 or 2 edges away. You wouldn't use anything but MISA. So the top 2 and apples were methods that existed before. So when we developed MISA, we were comparing it to existing things. So I'm not suggesting you should use the other methods. I'm suggesting if you have a mixture of 2 things, you should use MISA. It's just that this is like comparison to the existing methods to see how it works. Okay, so moving on, there is another cool application that we are going to go through during our tutorial. If you have a genome skim that's a mix of 2 species, is very similar to having a species that is actually a hybrid species, a very recent hybrid, right? And only if it's the alloploid, allopolyploid, say. If it's an allopolyploid hybrid, a recent allopolyploid hybrid, that's very much similar to having a mixture in terms of like once you get to the genome skims. And so we also tested for those yeast data sets that we have been looking at, there are a bunch of known mixtures. These are all known. And for each of them, we know what they are built, what the species they are hybrids of. We know that. So we took each of these and we tried to double place them on this phylogenetic tree of the yeasts. So these are your references in this case. When all of the constituents were in the data set, we had perfect accuracy with MISA. So it was able to find sort of the parents of these hybrids every single time. When one of the constituents were present but the other one was missing, it was also able to place the missing one correctly every single time. When both of the constituents were missing, it worked correctly for these 5 species down here. For one of the species, it was off by 2 edges. Like this species right here, you see how its constituents are sister to each other. When you remove both of these from your reference, the correct placement for this is going to be like both of constituents should be on this like short branch here. And MISA in that case was failing. It's putting one of them here, the other over there. But for the most part, the method is working fine. Okay, so I think that's the last result I have. I'm going to answer questions and then I'm going to move on to do a tutorial using this example. Is it known how polyploids that are in the process of depolyploidization will be depicted in such a case? No, the short answer is we don't know. I mean, that's an interesting question, a very interesting question. We haven't really modeled it mathematically or tested it empirically. So I won't be able to give you an answer, unfortunately. I just don't know. I don't even have a good intuition for what would happen. Maybe Metin, the developer of this algorithm, would have some intuition, but we'll have to wait to test that. So the other question is, how recent is recent? Basically, if it's if you're talking about hybridization that happens like millions of years ago, that's not recent. All these hybridizations that we are talking about are between the leaves of the tree. If you get to scenarios where hybridization is between sort of ancestral nodes, that is more complex, right? So for example, if you had a hybrid between, like, I don't know, this node over there and this node over here, I don't know if, like, we haven't tested under those conditions, and I wouldn't be surprised if MISA fails to be accurate in those cases. But maybe it's fine. I don't. We just haven't tested it. Like, the point I was trying to make here is that if it's a recent hybrid, it's exactly like a mixture of 2 species, right? It's sort of conceptually it's the same, but the algorithm might just work fine if the mixtures are not recent as well. We haven't tested it. Okay, questions? Any other questions? If we have, for example, new alloploid. Yeah, so if gene loss is just pseudogenization, so it depends. So when you talk about gene loss, if it's just like if the sequence is there but it's not a gene anymore, that's fine. That does not matter because these methods are not looking at genes, right? They're just looking at sequences. So if you lose a, I don't know, start codon and you don't have a gene anymore, or if the frame shifts and you don't have that gene anymore, that is not a problem because the sequence is still there. If like that chunk is actually removed, if the big chunk is actually removed, like 20% is removed, we have to actually test that. I don't want to speculate here. I honestly don't know what the impact would be. It's a good question, Andre, but I just don't know the answer. Okay, so I don't see any other questions, so let's do some hands-on stuff. Okay, let me get rid of all of these. Okay, so we are, this is a Thursday, so we are going over this tutorial here on your wiki. If you had opened it previously, please refresh. I updated just 5 minutes before the start of our session, so just refresh your page. So the tools we are going to talk about, the first one is MISA. There are some other tools we will touch on. These are just the references. Okay, so before we start anything, we need to get a compute node on our cluster. This is the command that we saw yesterday. So we are going to run that, and hopefully that works fast enough. Oh, I'm getting an error. That's not good. What is that error? Okay. Sorry, let me see. Maybe I'm running another thing on the background. That's no. Oh yeah. Okay. Sorry, I was testing something else, so I'm gonna have to cancel that. Okay, so let's try one more time. I had another interactive session. I think that's why it fell. No. Okay. Why is it not working? Anyone has any idea?
No, I don't know what's happening.
Okay, let me try from the other login now.
Yeah, I'm also having issues doing the same.
Okay, worst come to worst, I'm gonna just use the login node. We are usually not supposed to do that. So if that happens, I'm gonna ask others not to do the same, because if we all use the login node, the system is gonna be overwhelmed. But at least I can show you what happens. Let me try one more time. Okay, let me make sure I have nothing running. No, okay. Oops. Let's try one more time. Hopefully it works this time.
I even tried with other project now. It's a problem with the cluster. It's just giving a...
Oh, okay. So I think I had luck. So what I changed was the login node. I went from login node 4 to login node 1. I think that's what made the trick. I don't know. It worked. I'm not going to complain. So if for you guys, if it's not working, try logging into a different login node. You can do that by just going like SSH login dash, I don't know, 2 or something. And then you have to give the password. I don't want to do that right now. Login node 4 seems to not be working. Okay, anyway, so we got our node. One hurdle is overcome. Let's move on. Installation. So like everything else, we have already installed MISA for you. MISA is super easy to install. The instructions were part of like yesterday's instructions, so I didn't like repeat them here. On these machines, they are already installed. You just activate your this conda, and we are going to use FastME like yesterday again. So let's load that module. And we are also going to export this path so that we have access to some of those little scripts that I provided yesterday. Before the end of this workshop, you might want to copy this bin directory for yourself somewhere. It has all these scripts, although the link to them is in these tutorials. Okay, so let's go through this. We want to go to our user workspace. We don't want to overwhelm the home directories. And then this is the directory we created yesterday. I'm gonna create a new directory for our mixed queries. And this is one of those known mixtures. Let me show it on this slide. That's this green one. This green one right there is the one we are going to use as example query, right? This one. We are going to try to place it, and hopefully it's going to be placed as a sister to Cervacea and Eubionis, or whatever that name is, Eubionis. Okay, so we'll see if that happens, but this is going to be our query. Okay, so we just copy the genome. We start with working with genomes. We copy that genome to our mixed query. I missed something. Sorry, I should have said copy this thing to mixed query. Oh yeah. So there you go. We are copying it to mixed query over here. Okay, so now we have our mixed query directory there. Now, if you want to know what are the constituents of these species, we have them over here in this file. These are the two constituents that we should see. We saw that on this slide as well. Okay, so before we do this, I want to first go through a scenario where we do have both of the constituents in the mixture. Now, one of our two constituents, Cervacea, was our query yesterday, right? So this thing, if I look at the backbone tree that we had yesterday, it's not gonna have, where is our backbone tree? This one. It's not gonna have Cervacea in it. Let me display it. It doesn't have Cervacea in it, right? So we will get to an example where we will use this as backbone, but first I want to do a case where we do have both things in the backbone, right? So let's first do this. Let's update the distance matrix that we have. We have already added Cervacea to our library. Remember yesterday we used -A to add Cervacea to our library. It's just that we didn't update the tree. So let's just go ahead and do that. So I'm going to run this skimmer command to update the distance matrix to include Cervacea as well. It's going to take a minute. Once that's done, I'm going to just run this command to get a full tree that also includes Cervacea. So it has all the pure yeast in it. So when we see full.tree, we will know that it's the tree that has both of the constituents present. Let's give this a minute to finish. There you go. Okay. So now we are going to run fastme to get the full tree. So let's just look at the full tree, make sure nothing went wrong. There we go. Okay, so now we have Cervacea in the tree as well. So our two constituents, Cervacea and Eubionis, are both in the tree. Okay, so let's start analyzing the query. So the first thing that we should do is compute distances from our query to everything in the reference library. Remember, we do that using this skimmer query command. And we give it -T to do the Jukes-Cantor transformation. Okay, so we're gonna just run that. This is gonna take a minute. So what this is now doing is computing the distance from Pastorianus, which is our mixed sample, to all of the pure, to all the reference species. It shouldn't take a long time. It should finish soon enough. And once that's done, we are going to just run this little script to convert the format from one format to another. Remember, it was like row versus columns. Okay, so some people are still having problem with getting an interactive session. Yeah, try just logging into different login nodes, and that should hopefully work. Okay, so I'm just converting the distance from column to row. Let's look at the column one. Okay, so these are the distances from our species of interest to everything in the reference data set. Now, look at these and see how that theorem I talked about beautifully works here. Look at the distances. Let me make these look a little bit nicer. That's T, there you go. Look at these distances and look at the top two here. Cervaces and Eubianus. The two, I'm sure I'm butchering those names. Sorry about that. But anyways, you get the point. The two parents of this mixed species are the ones with the smallest distance. That's fantastic. So that sort of theorem worked exactly fine. The closest two species are the smallest distances. So right away, you kind of see an interesting pattern here, right? Those two distances are much smaller than any other patterns. In fact, in our paper, we computed what should this distance be. I don't remember the exact number, but it's like 1.6%, something like that. It's a fixed number, more or less. And the number that we are getting here is very similar to that. We could look up that paper and figure out that number should be. But when you see two very small numbers around like 1%, you know you have the right species. So this is this is not a fixed number. It is a number that is a function of the distance between the two constituents. It's a little formula. We have it in the paper. We could look it up. But if you know the distance between these two, you can compute what this value should be. Maybe if we have time, we'll do that later today. Okay, so any questions so far? Okay, I'm sorry, it seems to not be working for others. What I suggest you do is just follow what I'm doing, and maybe later you could try to go through these steps. Just email me if you had problems with these steps later. Okay, so that's good. So we saw that theorem in action. Now, the first thing I want to do is kind of like just ignore. So let's say we don't know that this is a mixture. We think it's a single species. And we go ahead and use apples, the tool that we used yesterday, to place this mixture on the tree. Sorry, I made some mistake here. I think my mistake is that instead of txt, I should give it tsv. I'm going to fix this. There you go. That not work. Okay. Let me make a note of this mistake that needs to be fixed in the tutorials. txt to tsv. I'll remember that later. Okay, so in this case, what we did was we ignored the fact that we have a mixture. We just used apple to do the placement, just like these are the same commands we saw yesterday, so I'm not going to describe them again. If you look at the tree, let me make it larger so that we can see this tree. or a species was added here as sister to one of the two constituents, Eubianus, with quite a bit of branch length, right? So Apples sees that this is not identical to this species. It also has like low distance to cervaces, but it doesn't know what to do with that. So it kind of like just puts it as sister to one of them. Now, in the example that I ran, I used this backbone tree without cervaces. Now let's use the full backbone that does have cervaces as well, and see what we get. Okay, so I just changed the output name, and we are gonna, we're going to convert the JSON to new tree, and we are gonna look at that new tree. Okay, so now we are placing on the tree that does have cervaces as well. And now look where Pastorianus goes, right? So it goes on a completely wrong placement. It kind of just puts them in between the two constituents, right? These are the two constituents. One of them is here, one of them is there. Apple finds itself very confused because our sample has low distance to this one, and it has low distance to this one. It can't kind of manage to make the distances work with one placement. So it just kind of gives up, puts it on this internal node. That's the best that it can do, and it's not good at all, right? So Apple wouldn't work in this case. You want to use apples only if you know you have a pure sample. Okay, so moving on. So let's now use MISA in this case. So we have already computed this distances, so we are good. Let me make a note for myself that these steps need to move up. My tutorial is a little bit not linear. Move up distance calculations. Okay. Then, so now we are going to run MISA. The command of MISA is very similar to Apple. We give it the distance matrix. Let me, before running it, before running it, let me show you what the inputs are. The inputs are this distance matrix, or I guess this distance vector, the distances from or mixed sample to each of the constituents, right? That's the distance vector we looked at in a column form. This is the row form of it. We also give it the full tree as backbone, and that's it. And then we tell it what the output name should be, right? And we call it mixed output present, meaning that both of the constituents are present. Now, when we ran Apple a second ago, it ran right away, right? It was like, it took no time. But for mixture analysis, you will see that it's a little bit slower, right? So it's now trying to, it's trying all placements. So it is taking a little bit of time. It has to do quite a bit of computations to be able to find the optimal placement. It won't be super slow, so don't worry, it's going to finish in a second. So let me look at the questions in the meantime. How does the proportion of the constituents of the mixtures would affect the placement? It is a threshold where it does not work anymore. Okay, that's a good question. I'll get to that in so two good questions. I'll talk about both of them in a second. Let me just, I won't forget, let me just show you the results. This run finished. So let's look at the results. So the results again is a JPlace file, just like we saw yesterday, is a JSON file. What is different about this JSON file is that now it gives you two placements. So Pastorianus 1 and Pastorianus 2, right? It gives you two places where this species is gonna go. It kind of added just this _1 _2. and basically it has just two placements, right? One on edge 2, another on edge 11. You can see that edge 2 is one of our constituent services, and edge 11 is the other constituent, Bionis. Now let's just look at it in UVic. UVic is more fun. So I'm gonna just convert this JSON file to NuVic. That's it. I'm gonna show first the full tree before we added the Pastranius, full tree after we added Pastranius. Let me just make this bigger. Okay, so you can't see the full tree before it, but that's fine. We look at it before. So this is the tree after placement, right? So you can see Pastranius is put as sister to Cervaces with a little bit of edge length. What does that little bit of edge length represent? The fact that, since the hybridization happened, there has been some time. So a little bit of evolution has happened. Same thing here. It's sister to Bionis with a little bit of edgeless, representing that, it's not just a mixture of those two species. It's like a hybrid with a little bit of evolution afterwards. Hope that made sense. Now let me stop here, take some, answer some of the questions while you guys are trying to digest what this results is. We are gonna get to the case where the constituents are missing, so the interesting case. Okay, so one question was, what if it's not 50-50? So this method does not care. This does not need the mixture to be 50-50. It doesn't actually matter what the mixture is in terms of like how much you have from each of them. So that's a good thing and a bad thing. The bad thing is that the method won't be able to tell you what's the proportion of each of them. Now, what it does assume is that the entire genome of both of the constituents are covered at least once. Now, if a little bit of it is not covered here and there, that's just going to be a small error, right? But you want to have coverage like 2x or something for both constituents, so that everything with high probability is covered. If you have 4x coverage, that's great. Now, so let's say you have 4x coverage of both of the genomes, but one of the, like at least 4x, but one of them is covered 16x, one of them is covered 4x. That's fine, it doesn't matter. It is not taking into account sort of multiplicities. So it doesn't care what the coverage is. It just needs the entire genome to be covered. So if you get to like, I don't know, half X coverage of one of the two constituents, it might start to not work, to answer the questions. Are you planning to expand to more than two constituents? That's our hope. We haven't done it yet. So what we have done and we haven't managed to publish yet is to increase the accuracy of MISA. We have MISA 2, which improves its accuracy even further. We haven't published that yet. Once we are done with that, we hope to get to a mixture of more than two. The problem keeps getting more and more complex. So we might have to like come up with a completely different kind of approach for a mixture of many things. Okay, so both are very good questions. Any other questions so far? Okay, so now let's get to the interesting case, right? So remember this backbone tree, it didn't have Cervaces in it, right? Cervaces is missing. So what happens if we try placement on this tree as the backbone? So let's run MISA and give it this tree as backbone and see what happens. So one of the two constituents are missing. Again, it's going to take a minute. I'll have a sip of my coffee and I'll finish. There you go. Okay. So our output was called this JSON file. We are going to convert that JSON file to a NuVec, just because we like looking at NuVec a little bit better, or at least I do. And we're going to display this NuVec before and so let's just do after placement. There you go. So this is what I've been waiting to show you guys. This is the cool thing. So let's see what happened when one of the constituents was missing. The one that was present, it worked as you expect, right? It just put it as sister to the one that was present. What happened for the one that was missing? So remember, right here we had that Cervaces species, right? It figured out that there must be some species that is missing here, and this would belong to that place, right? So it put it exactly where it would have put it if you had Cervaces in the mix, right? So let me just make this smaller so that we can show the tree where we did have both constituents present. Present tree. Okay, so I want to make sure they are both visible. They're not. Let me make things smaller a little bit. Maybe that works now. Okay, there you go. That worked. Okay, so so this is when one of the constituents up. Bottom is when both constituents were present. You can see that the results are basically the same. Like the fact that the Cervaces was removed on top didn't change your result, which is what we wanted, right? So it kind of figured out that one of the two constituents should be this far away from paradoxes, right? So it's not like putting it as very close sister to paradoxes, is putting it as very far away sister from paradoxes. If you were to take this tree and you remove Cervaces from it, you would get basically this tree, right? So the fact that we didn't have Cervaces did not confuse the method. Exactly. So that's still, even though one of the constituents was missing, is still correctly placing the mixture. That's kind of the magic of MISA. We had to go through a lot of like complex maths to get here, but at the end I think it's very exciting. I was very excited by this. Now let's try to make it a little bit more exciting. So let's, oh sorry, this picture is also out of place. I'll move that picture. It should be after the next example. Move picture down. Okay. Okay. So now let's work on a case where both of the constituents are missing. So I'm going to start with this. Oh, let me make things bigger so that you can see. Okay. So I'm going to start with this command. All it does is it's going to remove our other constituent from the backbone tree as well. So now we don't have neither one of the constituents are in the backbone. Both are missing, right? So we don't have Bionis, we don't have Cervaces. Both of them are missing. Let's go ahead and run MISA in this case and see what happens. And copy paste now. There you go. So we are copy pasting and running. Again, it's going to take a minute. So we'll sit back and watch it do its thing. Type in questions if there are any while this is running. If you have good Norwegian coffee, feel free to have a sip. It's going to finish in a second. There you go. Okay. So like before, I'm going to just change the format from JSON to NuVic and look at the NuVic output. Let me just make the whole thing smaller. I want to show both trees again. Okay, so before we look at it, let's also look at the tree where both constituents were present. There you go. Okay, so let's look at what we got here. Okay, so on top, I can make this a bit bigger. There you go. On top, we have the tree where both constituents were missing. Neither one was present. And still, it was able to find both placements correctly. Look at this. I mean, every time I see this, I get a little bit excited. And then on the bottom is when both constituents were present, right? So the answers are pretty much just identical, right? So we didn't know Eubianus, but we kind of figured out that Pastoranius has to belong in this place on the tree. And we didn't know where Cervasis was, but we kind of figured out that it should be in there. This is the magic of phylogenetic placement. You do not have to have references to be able to do identification to some degree, right? So like let's assume for a second that Cervasis was not known to science and Eubianus was not known to science, right? Or their genome was not present. If you take Pastoranius and placed it on the tree, you would look at this branch length and this branch length, and you would infer that, oh, this is a mixture, but it's a mixture of two things that I don't know about. It's a hybrid of two things that I do not know about. On the other hand, in this case, you look at this and you go like, well, this is a mixture of two things that are that I do know about, right? So it's a very kind of like interpretable kind of output. Sabrina is asking, but you still need some list of reference FASTA for this. Yes, not FASTA, but some reference, either genomes or genome skimming. Genome skims. Yes, you need something, right? You have to place it on some tree, right? But you don't have to have the same exact species in your reference. So when I say you don't need reference, that's what I mean. What I mean is that you do not need an exact match. You do not have to have that same exact species. You still have to have some context to place it into. Okay, so that's so basically that picture that you have in the tutorial is exactly what we are looking at in the terminal. It's just like a little bit more colorful and nice, right? When you actually this is for the case where one of them is present, so it was actually placed correctly. We now saw the case where both are missing as well. Let me stop here and take questions. If not, we'll do a quick rest. We'll take a quick break and we'll come back to the repeat section. Actually, you know what? So I'll be here answering questions if anyone needs to take rest. Why don't we start again at what do you guys think? 8:25. Would that work? Okay, so let's plan to start the live. In the meantime, I'll answer whatever questions you guys might have.
Hi, Siavash. That's
Hello?
really cool. I was wondering what happens if you do this multiple times, like if you have multiple allopolyploid data sets and you keep adding to this tree. Do you think some error might creep in?
So multiple times as in like you have multiple hybrids that you keep adding? Is that kind of what we're
That's right, yeah.
Okay. Let me think before I answer. I haven't thought about that. So your tree is growing, right?
Mm-hmm.
Good question.
I'm definitely going to try it out.
Yeah. So the way to try it is, let's see. I was gonna just try it right now, but I think, yeah. Okay. So before you can try that, we have to change something in MISA. So I can't try it right now. So I'll make a note of that. That's a very cool idea. We never thought about it.
And
MISA output constituent distance. So let me tell you what is MISA like. Let me tell you why you can try it right now, but it would be a very simple thing for us to add to the tool. I'm going to do it later this week. I'm going to ask Metin to do it. This tool is developed by Metin, who is a student in my lab, Metin Balaban. That's him. Okay, so let me just tell you what's missing. So remember I told you that we have a magic deconvolver that takes the mixture distances and deconvolves them to two matrices.
right.
That's basically MISA. Internally it does that, but it seems like it's not outputting these numbers. Like internally it has those numbers, it's just not like outputting them to a file or something.
Right.
Before we do what you suggested, we need to have these distances so that like before you can do the next round of additions, we need to be able to have these distances.
Mm-hmm.
And so we will have to update MISA to output those distances. Now, there is another thing that you're going to also need, and I have to think about it. I don't know. So you also need the distance between the two mixtures, right? And that is a little bit more complex, actually. I didn't think about that. Right. Yeah, it's like, so in the scenario where your mixtures are not present in the tree,
Mm-hmm.
we have a missing conceptual thing, which is given two mixtures, we will have to get like 4 of these vectors. So let's assume you have 2 of these mixtures. If you are adding both of them and you want the relationship between them, you have to do a like 4-way deconvolution, and that is not done right now. So yes, I don't think we can do that right now. But what we can do is to place them independently, right? So you can, and that is like completely doable, right? So like you could place multiple things on the same tree. You wouldn't get the relationship between hybrids, but you get the relationship between hybrids and the references. I know that's not probably what you were looking for. That can be done.
Yeah, but I think, so what I don't understand is if you do it only for one sample, the output of MISA is that, do you get a new distance matrix with everything in, or do you just get a tree?
You can get that. So internally it does compute the distance matrix. It's just not outputting it. We will change that in a second. Actually, you know what? Maybe it has an option to do it. Let me see. Run MISA health. Let me see if it has an option to output the distance matrix.
Because if it does that, then you can just do it iteratively.
No, that's what I first thought, but there is a problem, because it does, you can get all the distances, but then the next time around, when you have a new mixture, you can't get the distance from that mixture to each of these two constituents.
Yeah, sorry.
That's the thing that is missing. I don't know if that makes sense. Let me try to show you on this tree. So let's take this case, right? So I will have the distances from this point, like placement 1 and placement 2 to all the other species.
Mhm.
But now let's say I have a new mixture, right? I have a new mixture. I can compute the distances from this mixture to all the other species as input, right? But I don't know how to compute the distance from this mixture to constituent 1 of the other, like the previous mixture. This one, this right last arrow, I'm gonna just redo it in red. This red arrow, we don't know how to compute that yet, right? We don't know what that distance is. And we also don't know what this distance is. So that's why we won't be able to add the new mixture onto this tree, unless we can find a way to solve that problem, finding the distance of the mixture to both constituents of the previous mixture, if that makes sense. Yeah, that's a very interesting question. We have to think about it. But anyway, in the meantime, we will make sure the distances from your mixture to everything else is getting outputted.
Cool, thank you.
Any other questions?
I do have a question, but it's related to yesterday's session, actually. If that's possible, or you prefer me to leave it for the end of today?
Go ahead. We have 4 minutes in this break, so go ahead.
Yeah, I was thinking, I have this question because it's something you mentioned before about the influence of not having complete genomes, that they should be at least covered at least once. So I was thinking that I have a bunch of sequences that are not really low coverage sequences. They have like a 10x or 12x coverage, but these are kind of complex genomes. So the assembly is not that good. I have like 1.3 gigas out of the 3 or 3.5. So I was thinking of the influence of that factor on the placement or in the inference of the distances between the samples.
Yeah. So if you're using assemblies as input, that might become a problem. So there are several factors. If you're using assemblies as input, there are 2 scenarios. One is that all the assemblies are missing like pretty much the same positions. That would be okay, because the distance between the other areas is still going to be computed correctly. But if you have a case where, and I know it's like hard to know which one it is, right? But if you have a case where like one assembly is missing this part and has this other part, but the other assembly is missing, that part and has the, so if like they're non-overlapping, that becomes a bit of a problem. In that scenario, I wouldn't use assemblies. I would use the reads, the bag of reads as input. And I think that should work, except like if your genomes are super repetitive, we'll talk about the repeats in a second.
Yeah.
Then, we haven't tested this double placement with super repetitive genomes. So I don't want to say it works fine. I mean, you can run it, but whether the accuracy is high or not, it has to be tested.
Well, I don't have a double placement anyway. I was thinking of, just one sample trying to.
Oh, if it's single placement, I think it's fine. For single placement, you don't even need very high coverage. When I said, you need high coverage, it was for double placement. For single placement, low coverage works just like we saw yesterday. And repeats, we have ways to deal with it. I think for single placement, it should be okay. I wouldn't worry about it too much. But yeah, if your assemblies are very incomplete, just use reads directly.
Yeah.
What is preventing your assembler is repeated structures, right? But those are like the data is still in there in the reads.
Yeah. I know that there must be some more information in the reads, because there must be regions that are not assembled.
Yeah. You probably have some repeated structure in the genome, maybe segmental duplications or or genome duplication, things like that are prevented.
Yeah, but the thing is, I thought of this because I tried to run what we saw yesterday on my data today, but then I realized that the incompleteness of the genome. So I was thinking how accurate that inference might be. I did it on the assemblies because of it was faster and computational.
Yeah. No, if the assemblies are incomplete, I would rather run it on the SRA parts, on the reads.
Okay. Well,
okay, so it's 8:25. Let's move on to the next part, unless there is any other pressing questions. Why not do global alignment to identify whether assemblies differ in different places? Of course, that will not work good for highly repeated. Yeah, that's something that can be done. You can try to figure out if they are overlapping or not. Okay, so we're done with the code. So let's talk now about repeats now that we are talking about, it's a good segue to our next section, which is all about repeats. So when I talked about, genomic parameters yesterday, I kind of completely ignored repeats. At the very end, I kind of mentioned them. So, so the question that we're gonna now ask is, given a genome skim, so just a bag of reads with potentially low coverage, is it possible to, so previously we were able to estimate genome length and coverage, but can we also get some idea of the repeat structure of the genome? And I'll tell you in a second what we mean by that. Knowing this repeat spectrum is going to be really helpful for getting more accurate estimates of the genome length and coverage, which in turn can help distance estimation. There are some methods out there that try to estimate repeat structure and genome length from genome skimming data, so basically just a bag of reads. But they weren't really focused on super low coverage, like 1x or 2x. They assumed you have high coverage, like I don't know, 10x, 12x, something like that. So what we are going to try to improve compared to the existing methods is enabling very low coverage. Okay, so what do we mean by repeat structure? We just mean the repeat spectrum. So this is, I'm pretty sure you guys have already covered this in other talks, but just to make sure we are on the same page, basically the output that we want is something like this, where we basically say these many of the k-mers in the genome are not repeated at all. They are observed once. These many are repeated twice, 3 times, all the way to, I don't know, 1,000 times, right? So this histogram is what we are talking about when we talk about repeat spectra. Now, obtaining this spectrum from your reads is trivial. You just count how many times you have reads and you get this. What is difficult is that spectrum is not going to be just a function of your genome's repeat, but also coverage and error and all sorts of factors. What we want to get is the repeat spectrum of the genome itself, given the repeat spectra for the genome skim. So that's why this is a tough challenge. Now, if you have it, if you have this repeat spectrum, your genome length is basically like the y-axis multiplied by the x-axis summed over all values, right? How many, so this is, sorry, how many times you have seen something, so how many of the k-mers are repeated once? How many of the k-mers are repeated twice? How many of the k-mers are repeated 3 times? If you sum those up from 1 to, I don't know, infinity or whatever, you get the genome length, right? So if each position in the genome is in one of these like bars. I hope that makes sense. Okay, so again, if you have the assemblies, computing these repeated spectrums is trivial, right? You just count how many times you have each k-mer. A tool like Jellyfish would be able to compute that for you. It's a very nice and optimized tool I suggested. So we took 600 assemblies, 622 assemblies from RefSeq. Some were invertebrates, some were plants, some were mammalian, and some were other vertebrates. And we computed these repeat spectra, and we are just looking at basically what portion of the genome is seen only once. So R1 divided by L. What portion of the k-mers in the genome are seen only once? And our K, I think, is 31 here, or 32, I forgot. And what you see is what you expect. So plants, which are known to be very repetitive and whole genome duplications and things like that, they are, this ratio is very low for them. So there are some plants where like less than half of the genome is seen only once. And if you have recent genome duplications, that shouldn't be surprising, right? If you look at mammals that are less repetitive, things look better, as in like there are more unique parts. And when you look at invertebrates, it's a mixed bag. There are things that are like super repeat, like very free of repeats, and there are some things that are very repetitive. We have all sorts of things. Okay, so this is just a demonstration, but let's say you have a genome skim, you want to know how repetitive it is. Can't we just find the closest genome and compute it for that closest genome and call it a day and be happy with that? So the answer is unfortunately no. This is a tree of I think it's just a taxonomy of plants, a bunch of plants that we had in our test set. Now let's look at it. So the color of these labels is basically how repetitive. So R1 divided by L. What percentage of the genome is repeat free? Now there are some clades where everything is similar, right? So if you know one of them, you can look at these close things, and they'll all be similar. But then you also have things like this where a bunch of relatively similar species have very, very different repeat structures. These are plants that have been, some of this is because of domestication, right? But this is not like the only example. There are other things like, let's take like, maybe I should have highlighted it, I forgot. There's also like some genus here where the two species in the same genus have quite a different repeat structure. So you can't just look at the closest thing. I mean, that's not a bad start, but it's not sufficient. Okay, so now let's think about this a little bit more mathematically. So let us assume that R, this vector R here, is the repeat spectrum of the genome. So R is what we are interested in. R is what we want. M is what we observe. M is the repeat spectrum of the genome skim. The repeat spectrum of the genome skim is the function of the repeat spectrum of the genome itself, of course, but it's also a function of the coverage, and error, right? So P, this matrix P here is kind of like, what's the probability that you would sample a position with a certain coverage given its repeat value? And it's also a function of error, right? So both error and coverage impact what we observe. And what we want is just R itself. So we want to get rid of coverage and error. So how do we do that? Well, just like before, we have to come up with a smart algorithm to compute coverage and error, and also genome length, which we don't know. Once we know those, then we can kind of translate from M to R, right? But because we don't know these, we have to kind of do all these estimations together. The math of it actually gets really complicated really quickly. And there are like some negative results, meaning that not everything is always you can't always estimate everything with high accuracy, but basically you can do some sort of like iterative approach. You assume some value for these parameters, you do the convolution, then you assume some value for other parameters, and then you kind of like solve this problem. You keep going back and forth. Even that is not enough. You have to add some other nuts and bolts to this algorithm. I don't want to like spend a lot of time on how the algorithm works. So I just hope the model makes some sense. How we solve this, that's a lot more complex. So let's not worry about that right now. Just know that we do have a solution. It's called Respect. Again, like everything else, it's available on GitHub. The paper is on BioRxiv. It's going to be published any day now. It's going to be published in PLOS Computational Biology. Okay, so let's look at some results. So these are genome skims that we simulated at 1x coverage, so not very high coverage. And we have 1% sequencing error, so pretty high levels of sequencing error. This method here is the previous method that existed before we before we developed Respect. Y-axis here is the error in or estimate of the genome length. So remember yesterday we had some error in estimation of genome length, like we had about, I think, 7% error in our genome length estimation. Once you use Respect, the genome length estimation error goes down a lot. This axis is logarithmic. So the vast majority of cases or genome length is estimated within 10% of the correct value with only 1x coverage. So that's quite a bit of improvement. And this previous method called CovS wasn't quite as good. And in fact, it was quite a bit worse. Most of the time it was more than 10% error. And it's not surprising. It wasn't really designed for such low coverage. Okay, so that's first results. So these are the averages, 4% error versus 40%. Let's look at a bunch of these are still simulated genome skims. Again, this on top, we have this existing method, CovS, and in blue, we have the error of OR method, Respect. And you can see that there are like 3 or 4 cases down here where our method fails to have very low error. It has like, I don't know, 10% error or higher for these 2 cases. These are super repetitive genomes as well. But there are also some super repetitive genomes where it works exactly, completely fine. So that's another example. In less than, in more than 90% of cases, the error is less than 55%. So basically, it works very well. Again, there are some cases. So these are now real genome skims. Previously, we were using art to simulate genome skims. Here, we are using actual genome skims. And again, results are good, except like these 3 cases. There are 3 genome skims here where the method has error above 30% in its estimate of genome length. And we think those are cases where the genome skim has a lot of contamination that we haven't detected. Those 3 right there. There is another one, we didn't find contamination evidence for this one, but also we have 15% error. We are not sure what's going on. But look at all these green ones. These green ones, the 1, 2, 3, 4, these are cases where the genome is extremely repetitive. So only like 30% of the genome is repeat-free. 70% of it is repetitive in this case, and all of them are super repetitive. if you look at, for all of them, assembly is also available. This is how we know the, or error, right? So the assembly length was 4.3, we estimated to be 3.9 with just like genome skims, and our estimates is pretty good, right? And you know, for all these cases, you can look at the assembly value and our estimate. Our estimate is sometimes a bit higher than assembly, sometimes it's a little bit lower, but it's close. And you know, this existing method wasn't quite as good. Okay, so I'm gonna stop here, answer some questions, and then we'll do a tutorial. What is the max genome size? So yeah, so I mean, in this case, I mean, we just took everything that we could find across the tree of life. So we do have 4.3 gigabase per. I mean, the better question, I think, is what is the smallest one we have looked at? And I don't exactly know, but we do have invertebrates. So hopefully some of those are smaller. How if the genome is not super repetitive? Is there something we should caution about in such cases? No, if the genome is not very repetitive, like just running a skimmer alone would be fine. It will give you a very good estimate of the genome length. A skimmer, which we looked at yesterday, start to be inaccurate for these kind of super repetitive genomes. So I wish I had one of these in my tutorial. I don't. Maybe I'll add it for the next time. If you were to run a skimmer on a genome like this that is super repetitive, the genome length is going to be underestimated. Not as much as this other method, but it's going to be underestimated. If you don't have repeats, it's fine. Any other questions before we jump to tutorial? Okay, there. So respect out of the tools that I'm going to show, what is about extra large genomes and more and ultra small ones? So I doubt we have tested on anything like as large as 30 gigabase pairs. I don't see why it wouldn't work. I think it should still work. Like conceptually, I see no reason for it not to work, except we haven't. I doubt we have tested on anything that big. In terms of ultra small ones, if you're talking about like, if you're talking about like bacterial level, again, we haven't tested. I want to say this still works, but I'm not sure at all. I'm not sure at all for small ones. For bigger ones, I'm a little bit more optimistic that it actually would work. So, you know, we can still run the method, whether it has high accuracy or not. I'm always like I don't want to say it works unless I have tested it, and you know that's why I'm a little bit hesitant to answer. Yeah, something like you know, this like super large and repetitive genomes would be interesting. So you know what? Let me make a note of that. That's a nice thing to say. That was, I'm gonna remove, forget this. That's a lot, you know. Has it been designed with big genomes in mind? Yes. Non-bacterial genomes. We haven't tested it on bacteria ever. It's been designed with eukaryote genomes in mind. So when I say big, I don't mean like 1 gigabase or something necessarily, right? It would work for those, but it would also work like our test case has a lot of these invertebrate genomes that are smaller, that are relatively small. They are like hundreds of megabase pairs. So it works on those as well. But once you get to like 1 megabase pair, that's the case we didn't have in mind and we haven't tested. So for eukaryotes, let's say. Do you have any comment in your estimate of heterozygosity? So heterozygosity, no. Heterozygosity respect, yeah. I don't actually. I don't. No. Yeah. So heterozygosity is more difficult to estimate. Of course, heterozygosity is going to sort of change the repeat spectra, but it's not something we have tested super carefully. So I want to like hold on. So respect is a very new method. Like I say, it hasn't fully, it's going to be published soon. So there are a lot of questions I'm going to say I don't know, unfortunately, but these are all good questions because they give us something to think about for next versions. But also several exogens for gymnosperms might be good to test. Okay, that's good. I guess I don't want to give me heterozygous. I was just wondering. Okay, so I'll have to get back to you about the heterozygosity. I just don't remember us ever testing that carefully. Okay, so for respect, like I just said, it's a new method. Unfortunately, it's a little bit more clunky than our other methods in terms of installation. So bear with me in terms of installation. There is one problem with respect in terms of installation, which is it uses this other tool called GroBPy or Groby, basically, which requires a license. It's not a free tool. Now, for academic use, we get a free license, so that's what we are going to use today. But there is a problem when you try to use that on the cluster, because whatever node you go to is like, oh, I haven't seen this node before. I need a new license. So I have to do like some extra stuff here on this new node that we have here. Do we still have the node? Yes, we have this node. So I have like some instructions in the tutorial. I apologize for the, we are planning to fix this, not use Groby anymore. If you are doing academic work, you can come to this Groby website and get free licenses. If I'm guessing you haven't done that in the past, so you could just see what I do and maybe not, like if you haven't done it in the past, just skip the steps for now and just look at what I'll do, and later you can figure it out. Okay, so I have to get a new license and I have to remember how to do it. Because again, whenever you change the node, unfortunately the license stops working. Okay. Oh. Right, so I have this, I have 4 licenses so far. Soon enough, they're gonna stop giving me licenses. If I had this node, no, I have a different node. I didn't get lucky. If I was lucky and I had the same node, I could reuse my license. Okay, so let me see if I can get a new one. How do I get a new one? There you go, academic license. You tell them you're a good person, you are not using it for something else. For some weird reason, this is always empty the first time you look at it. If you just refresh it, it tells you, oh, okay, this time that didn't work. So let me do a different thing. I'm gonna go here, hopefully it lists it. Okay, so I got a new license. What's the most date issued? 14, 15, 16. Okay. So I'm gonna take this license and activate it. This is the my license number. So let's get it activated. So I have the instructions for activating the license here. There it is. So that's how I will activate that license. Oh, okay. Okay, doc, and I need to give it that key number that we got on that website. There you go, this thing. And hopefully that works. Actually, before I do this, let me go through the other steps. So we have our license, we can activate it any second, but I want to go through the other steps first. So we are going to, so installation is already installed. You just have to load two modules, Jellyfish and Sequence Toolkit. So let's load our first one didn't work. And the second one didn't work as well. Okay, so we're gonna try one more time. We have to swap GCC. So we have a different version of GCC. We have to swap it. That usually gets us down a rabbit hole that we have to unfortunately go to. There is nothing else we can do. It keeps saying, oh, first swap this other thing. Once you swap that, once you want to swap that, it tells you, oh, no, first swap this other thing. Okay, there you go. Okay, so now that worked. Okay, I can now do this one. No, okay, another swap. There's probably a better way to do it. I just don't know what it is. Trust me, this is not as bad as it looks. It's gonna finish. That worked. Okay, that worked. Now let's do this one. That seems to work. Let's do this one. Okay, so now we can go back to this. Okay, I think that worked. Okay, so we went from one GCC to another. All of that was just doing that. Okay, now we have Jellyfish loaded. We have sequence tools loaded in a second. Good. Okay, so this is already installed, so I don't need to install it. The rest is just installing, which is already done here. And because we have our Conda activated, it's fine. Okay, so now I'm going to prepare input for the respect. So I'm going to make a directory respect, go inside it, and create a directory called inputs. What I'm going to do is put all those like yeast genomes. I'm going to just link them. So I'm going to link both. Oops, sorry. Okay. Okay, I did this the wrong way. Okay, so I have to first go inside input, then make these links and make sure my commands are correct. I think my command has a small error here. It shouldn't have this. Okay, I'm gonna fix this in the tutorial. There you go, okay. Okay, so I just linked to both FASTQ files and the genome assemblies for all those yeast genomes inside our input directory, right? So that's what we have inside input, just a link. I didn't copy them, I just linked to them. So we're gonna go back here. Okay, so let me see what's in the chat. I can run this web just for non-production, but it runs well. Okay, great. So maybe this license thing wasn't needed. Anyway, for me, I had to do it, but I don't know. Maybe it wasn't necessary. Maybe I was worrying too much about it. Okay, so before running respect, in my case, because I already have installed these licenses once, I can't not do it. You guys might be able to get away with not doing the licenses. Once you install it once, you have to do it on every new node. So I am going to activate my license, which hopefully works. In which directory do you, okay, I want it to be in the current directory because the one on the home is for login node. Okay, and now I need to just export this thing. So I'm gonna just do export. Oh, we are all set. We are ready to remove to run respect. Again, the licensing I just did, you guys might not have to do it. Thanks, Sabrina, for pointing that out. Okay, let's go ahead and run respect. So once you do all of that, now running it is easy. You just give it the input directory where you have all your genomes and genome skims. By default, it's going to do 1,000 rounds. It's going to take 1,000 rounds, takes a little bit of time. So I just reduced the number of rounds to 100 just so that we can get the result faster and look at it. For your real analysis, don't reduce it. The number of rounds, the more you do, it's like the rounds of those like going back and forth between the two optimizations. The more you do, the better, right? So don't reduce that. But for now, I reduced it so that we get results in, I don't know, 5 minutes or something. And we can move on and look at the results. Okay, so while this is running, let me try to answer some questions. Any questions about either installation or anything else related to respect? So if not, I'll just tell you what it's doing as best as I can tell. So first, it's running Jellyfish internally to compute the k-mer frequencies. It's doing that both for like assemblies and for the genome skims. Like whenever you see FQ, you know it's a FASTQ file, so it's a genome skims. When they end with FNA, they are the assemblies. It's right now just computing the actual histograms. It's not even starting the actual run. Yes, Sabrina. So Sabrina is asking, is the uniqueness ratio R1 divided by L? Yes, that's exactly what it is. R1 is how many of the k-mers have been observed exactly once, and L is the genome length. So that ratio R1 divided by L would be your uniqueness. Okay, so it's done. Oh no, no, that's fine. Okay, I thought it's not finding. Okay, so it's done computing the histograms. Now it's actually running the algorithm, the iterative algorithm that tries to separate out, that equation we saw, right? So now it's gotten to that point where it has computed M. Up until now, it was computing M. Now it's trying to kind of like solve this equation and compute R, given the fact that you have M, right? And to do that, it has to iterate between estimating all these parameters one at a time. So it's going to do 100 iterations because we gave it dash n 100. It's not that bad. Do you run Jellyfish with limited set to 10,000? No, I don't believe so. I don't think we do that. I think we do Jellyfish to get all. Oh, do you mean like maximum? Wait, limited set? No. So I'm not exactly sure what you mean by that. If you mean like limited number of k-mers, no. If you mean limited like maximum repeat, then the answer is yes, and that's something that you can. Okay, so respect. Oops, I haven't activated Conda. Let me activate Conda. Oh, okay. Let me activate Conda. There you go. So this is a different login node. Okay. So when you run respect with -h, it tells you what options it has, of course. One of those options is -n. It's basically saying in that histogram, what's the maximum value you go up to? So this goes to up to 50, right? So then by default, you can of course change that. So by default, by default, it goes R1, R2, R3, all the way up to R50. But the number of k-mers it's considering is not limited to 10,000. It's considering all of them. That's why it takes a little bit of time. Also, by default, the maximum or the minimum value for R1, the uniqueness ratio is 0.1. It has all sorts of other parameters that are less, these have to do with like nitty-gritty parts of the algorithm. You might, they may not mean much to you, so you can kind of ignore them. The important one is this -n and capital. And capital N, the higher this number is, the more it's going to take. We reduced it to 100. And K, I'm sure is important as well. We just use 31 by default. There are a bunch of options about what to take as input, what to take as output. Some of these just have to do with mapping of names and things like that. If you have already run jellyfish and you have the histograms, you don't want respect to run them, you can tell it. And there is all like this other option, which let me see. Okay, so it's still running. So let me tell you what this option is. When you get the output from the from respect, like even though it went all the way to R equals 50, it's just going to output the first 5, because after a while it becomes very noisy. So we decided to really focus on the top 5. Everything after 5 is going to be added to bin 5 by default. Now that number you can change. You can ask respect to output all the 50 numbers if you want. And if you have increased that number 50 with -n, you can also do more here. So this option is also kind of important. But just like having the top 5 makes it easier to look at the data. So I just went with that for now. These other options are, again, just like how you do input, output, nothing exciting. You can just read through them and hopefully they would make sense. Okay, and then you have a bunch of other options for temporary files and whether you you want to be able to work with zip files or not, things like that. Nothing super important. You can just read through them. Okay, so it did finish. It take me, okay, so something bad happened here. Oh, no. So when I see this error, it usually is because the license didn't work. Which is surprising me because it seems like yesterday this was working. Let's see. Yeah, this error usually means the license is not working. So let me see if we got an output anyway. Now they can always just add local file handler. Something is wrong with my installation. I'm not sure why. Sorry, this was not happening yesterday. Let's look at maybe it worked fine. Let's just look at the results. Okay. If not, we will try to figure out what went wrong. Yeah, this did not work for FAQ files. Give me a minute to see if I can guess what happened. Where we are happened. Zip import error. It's having trouble with. Oh, did this somehow get removed? I'm guessing. Maybe it's because multiple people are trying to run this somehow. That's not my username. I know what happened. So someone tried to install Respect in this that I was running on, and it really confused it. Okay, so I hope you guys understood what happened. So one of you guys tried to install Respect, given the instruction I had given you, but you were in my condo environment, so that messed up my installation, and that's what stopped it from running. So what I have to do is to reinstall it again. In the meantime, please don't try to install Respect unless you have your own Conda. If you are using my Conda environment, let's not do it. Okay, let's do this one more time. Okay, so I think this time it's gonna work. So I'm gonna run it one more time. Okay, so no one else should install anything right now while we are running this. Sorry, I should have mentioned this earlier. Okay, where is it? Oh, it's right here. Okay, so we're gonna run it one. This is a node, yeah. Run it one more time. There you go. Okay, we're gonna run it one more time. In the meantime, I don't want us to all wait another 10 minutes for this to finish, or I guess 5 minutes, whatever it is, for this to finish. So what I'm gonna do is I have results saved from my, no reason to be sorry, Paolo. I should have mentioned that, I forgot. Don't worry about it. Okay, so, but the good news is I have the results from my runs yesterday when I was testing this. Yes, Respect back, there you go. I kept those. So we're gonna, you're gonna just trust me that if that run finishes, we're gonna get similar results to these. Let me just make sure I kept the right thing. Yes, okay, so just trust me that you're going to get these results when that run finishes. In the meantime, let's just go ahead and not waste time and look at the results. Okay, so I'm going to look at the results with this command. So this is the file. I just like to make it look nicer. That's all this command does, nothing fancy. Okay, so what we did is, let me make this larger so that you all can read it. Okay, so what we have here is basically FNA files are assemblies, and FQ files are the genome skims, right? Or 2x genome coverage genome skims that we simulated yesterday. So ideally, so the assembly results, there is no our algorithm is not doing anything. It's just running jellyfish. It's giving you the it's giving you the profile for assemblies, right? So those are kind of ground truth. Whatever you have for assemblies, that's the actual value. And then hopefully what we get for genome skims matches the assemblies as well. Now, this run that I have here is probably with N10. So we'll see once the other one finishes, hopefully the accuracy is better than this. But let's look at this. Yeah, this is N10 because the accuracy is actually not super high. So for example, for this one, we are getting uniqueness to be 1, where it's actually 97. For this one, we are getting 87. whereas the actual accuracy, so this is the uniqueness ratio. We're getting 87, where the real value is 97. The reason these numbers are not so good, I think, is because I ran it with -n10 in my test case so that it finishes fast. And so we're going to look at the results once the actual run finishes. This column right here is your estimate of the coverage. The true coverage was 2, right? And you can see that these are also, some of them are a little bit off. This is the sequencing error. That's okay. It's fine. HCRM is, I actually forgot the definition. So let's go to respect and see if we can find it out. Respect, GitHub, Shahab. Hopefully it's described there. I think it has HCRM. Okay, so basically is the average multiplicity of k-mers with the highest multiplicities. Now, I don't remember exactly what's the definition of highest. We can try to look that up in the help. But it's basically saying for k-mers that have high multiplicity, and again, I think high, I forgot what it is. It's probably like more than 5 or 10 or something. What is their average, the average number of times that you have observed them? And it's divided by the genome length, divided by 1,000,000, right? So basically, like, how many high copy repeats you have per 1,000,000 base pairs. That's the way to interpret it. So like if you have, it's per 1,000,000 base pairs. So if you have like 7.37, means you have 7 high copy k-mers per 1,000,000 base pairs. Now, what is exactly high copy of? Unfortunately, I don't remember. So let's see if the help tells us. Yeah, unfortunately, it's not. I have to ask Shahab to update the help to mention that. Yeah, I don't remember exactly what we meant by high multiplicity. So let me make sure we will add that. High multiplicity exact definition. But basically, so I mean, sorry, the exact definition I don't know, but conceptually it means these are the number of very highly repeated k-mers that you have per 1,000,000 base pair. Hope that made some sense. So if you have 13, that's what it means. Okay, so let's hope this will. Okay, this finished this time. Okay, so with the correct installation, it finished. It took 5 minutes. A little bit more than 5 minutes. It took 5 minutes after we were done with like histogram. If this N100 was 1,000, it would have taken 50 minutes, so 50, and you would get more accurate results. But let's look at the results that we got here, and hopefully they are more accurate than those old ones that I get with N10. Oh, sorry. So it doesn't overwrite the previous results. It creates a new file with _1. because I ran it once unsuccessfully and I didn't remove the file. We have to look at this _1 files. Okay, good. Now, see, now our estimates are a lot better, right? When we went from N10 to N100, our results got a lot better. So look at the first one. The true uniqueness ratio is 97. We are estimating to be 98. Now, unfortunately, this yeast data set is not the best example. Actually, not a very repetitive genome. Maybe we'll run it on something else later if we have time today. I guess this is the lowest one. The true value is 94. We estimate it to be 95. Genome lengths are super close, right? Everywhere genome lengths are getting estimated correctly. And the coverage is very close to 2 now, which is the correct coverage value. So with N100, things are working pretty well. With N1000 default, it will even work better. I do have some Drosophila genomes lying around somewhere. Genome skims, sorry, not genome. So let me make a link to my Drosophila genomes and start that run. We will go to the next part of the tutorial while this is running for Drosophilas. Okay, so this is where do we have those? Okay, so let's see. Where did I put those Drosophila genomes? Not here. Drosophilas are going to be more. Okay, there you go. That's the location. Let me make a link to Drosophilas. I'm going to start respect on Drosophila genome skims, and we'll go about the rest of our. Let's also do and I don't know 200 or something. We'll continue the or talk while this is running in the background. We'll come and look at it later. Okay, any questions on respect before I go to contamination removal, the last part? We have 40 minutes. I'll try to get as much as possible in about the contamination removal, so I'll be a little bit faster. Any questions? No, okay, so, right, so skimmer, okay, so Samuel is asking a very good question. How do we combine these results with the skimmer? That part we haven't published yet, but I'm going to, there's a script to sort of there's a version of a skimmer that can take as input the output of respect. Let me make a note for myself. I'm going to make that available. Just be aware that that combination hasn't been published yet, but like I showed you, it's higher accuracy. So make a skimmer plus respect available. I'm going to make that available to you guys later this week and send add it to the wiki. Thanks, Samuel, for reminding me. Okay, so contamination, right? If you have reads after you do BB tools, you have to run you have to deal with contamination. What do we mean by that? When you have a genome skim, it's usually it includes your species of interest, DNA from your species of interest, but also other things. It almost always includes a little bit of human. Human contamination is really hard to avoid. It might have it definitely has bacteria. Well, most cases it has bacteria, but also if there are symbiotic species that live together, you might get a little bit of those as well. So it doesn't have to be just bacteria, right? Other kinds of symbiotic species. How do you remove them? Well, the first type is human contamination. That's easy to deal with, right? You have your genome scan, you take all the reads and you just try to map them to the human genome. If they map to human genome, you call it a contamination, you remove it, you are done. This works fine unless, of course, you're working on primate genome skims or something like that. Then it's not so easy, right? But for most everyone else, it's going to work fine. So let's not worry about human contamination yet, just because it's easy to get rid of. Another case that is easy to deal with again is if you have a closed reference genome for your genome skim, right? So, and by close, I mean something like 5% different or something, right? So if I have a genome skim and I have a genome assembly, let's say from the same genus, for example, right? Or sometimes, the same maybe family would work. Depends on their distance. You take your reads in your genome skim and you use BLAST or Bowtie or any read mapping tool that you like. If a read maps to that reference, it's fine. You keep it. If it doesn't, you get rid of them, right? If the reference is close enough, this would work. If you don't have a close reference, what do you do then? That's the most interesting case, the most challenging case, right? In that case, I think your best bet is, so I don't want to claim we have like full answers here. We have some answers, they work pretty well, but this is an ongoing like research question in my mind. Your best bet is trying to remove contaminants. And what do we mean by that? So what we mean is like, what kind of contaminants do we expect to have? Let's say bacteria, usually like fungal species are contaminants. If you have plants, human, we already talked about. So if you have some idea of what kind of contaminants you might have, you can try to look for those contaminants and remove those. What's the challenge? The challenge is that like that specific species of bacteria that is a contaminant, you don't even know which one it is. You just know bacteria are going to be contaminants. And even if you knew it, you might not have the genome, right? So that's one issue. You do not know exactly what, or you might have a general idea like fungal species or bacteria, right? At that level, you might know. And then the other issue is, of course, if you want to blast versus every possible species out there, it's going to be slow. So how do we solve these two problems in one go? Now, before I solve them, let's actually ask a simpler question. Are skimmer distances sensitive to contamination or not? Maybe a skimmer doesn't care, like it's not sensitive to contamination. So we did some simulations again. We have genome skim 1, genome skim 2. In each case, we have the main species, which is called A, and they have like some Jacquard, right? So they have some like level of similarity in terms of their K-mer set. We also add contaminants from one other species. And these contaminants also have some similarity to each other, right? Like the contaminants are not 100% separate between your samples. So they have some similarity and some differences. The contaminants are not exactly the same, but they are also not exactly different. So we are going to use the term H for the Jacquard index of the contaminants. And we continue to use J for the Jacquard index of like our actual samples. The Jacquard that we observe is going to be kind of like the summation of the two Jacquards, right? And we are going to estimate the distance between these observed Jacquard instead of the true Jacquard, okay? But is that distance going to be any good? So that's sort of like the results of our calculations and simulations. So what are we showing here? X-axis is the amount of contamination. So how much of our sample is A, how much of it is B, right? At the maximum level here is 50-50. At that point, your sample is hopeless, right? If 50% of your sample is contamination, it's, you throw that out and you start from scratch. But you go from all the way from 0 to 50%. And the different panels are different true distances between like A1, between your two samples, right? So they are your like actual distance. We go from two things that are very similar to two things that are quite different, 18% difference. And at the y-axis here is the error in the estimation. So 0 is good. When we are above the 0 line, we are overestimating the distance. When we are below the line, we are underestimating the distance. And the colors are aged, so similarity between the two contaminants. How similar are the contaminants? When it's dark, the two contaminants have nothing to do with each other. When it's light, the contaminants are exactly the same. You have the same thing contaminating both samples. Okay, so what do we see? What we see is that if your distance is, let's say, large enough, 2% or higher in these scenarios, and if your contamination level is low, let's say 20% or lower, these errors in estimation are not super high. The errors in estimation do get very high when the distances are very small or contaminants are very similar to each other, or levels of contaminations are very high. So there is some robustness to contamination. It's not like these levels of error shoot up very fast, but contamination can become a problem. That's the bottom line of this figure. Let's look at it actually in a more, we will see more interpretable results in a second. Andre, I get to your question later. So now I'm going to ask, can we remove contamination using existing methods? The best method to do this kind of thing prior to our work was Kraken. Kraken is a great tool. People who do metagenomic use it all the time. It's also another k-mer-based method. I don't know if you guys learned about it in this workshop. If not, try to learn about it. Among the k-mer methods is one of my favorites. What it can do is you give it a large database of many genomes. You can give it like tens of thousands, maybe even hundreds of thousands of genomes, bacterial genomes. I'm talking about bacterial genomes now. And it can search each read against those and try to tell you if, like, which of those species or groups of species in your reference the read belongs to. Now, we don't care about that. All we care about is whether the read belongs to any of these bacteria. So at the highest level, at the domain level, does the read belong to the domain of bacteria or archaea, or does it not? That's what we care about, because we know bacteria and archaea are going to be contaminants. We want to remove them. We could do the same exact thing with fungal species or whatever other kind of contamination you expect to have. The tool is pretty fast. It takes 100 seconds with one core for 1 gigabase pairs of queries. So that's pretty good. What it does is basically it finds whether each read has k-mer matches to anything in the reference above like a certain number, above a certain percentage. So by default, that percentage is 0. So when we say above 0, it means if any of its k-mers matches the database. That's the default setting. So we wanted to test this default setting for contamination removal. Now, the thing is, what makes this problem is difficult is if you have contaminants that don't have a close match in the reference database, right? So we tried to simulate that. We created contaminants that have an exact match in the reference database. That's this like 0 right here, or the ones that have a match in the reference database that of distance between 0 and 1%, 1 to 2%, 2 to 3%, all the way to, you know, contaminants that where your closest match in the reference database is more than 25% distant. You know, bacteria is a large group, right? So you can easily find bacteria that do not have a match of even 25% distant in anything in your reference library, even if your reference library is very big. Or reference library here was from like this tree of life database that we had done before. It was supposed to capture as much diversity as possible, but it's still, some things are not even like in 25% distant to anything in the reference database. Okay, so how well does Kraken do in finding contaminants? The bottom line is that it works very well if you have a close match. And what do we mean by close match? Up to 5%. It actually works decently well if you have like 5 to 8% distance as well. It finds most of those contaminants. But if you have contaminants that are far enough from your for your species of interest, let's say from your reference database, it's gonna fail to find them very often, right? So it's not so good if your contaminant doesn't match the database at close distance. This should not be a surprise. I think the number is constructive, right? You need like 5 to 10% similarity to something in the reference for it to work. Now, Kraken has all sorts of other sort of settings that you can play around with. Maybe those would increase the accuracy. We tested those, they don't. There was one setting that we found that increased the recall, but it also increased the false positive rates. So false positive rate here is how many things that are not bacteria is it going to classify as bacteria. All of these have decent false positive rates, so you don't get rid of your real genome skimming reads. But if you go to this setting when the recall is good, you're going to remove your real like species of interest. So you don't want to use this setting with high false positive rate. And among the other settings, none of them are any better than the default setting. So we're going to just stick with the default setting for Kraken. Okay, I mean, this is pretty good, but can we do better than this? Can we increase the matching percentage in these challenging cases? That's the question we asked ourselves. And the answer is that yes, you can do a little bit better. Let me see how I'm doing in terms of time. Okay, I have something like 30 minutes. I want to try to do some demonstration. So let me try to go a little bit faster here. Okay, so back to some data. So these are like genome skims where we sparked in some bacterial contaminant all the way to 25%. And we asked what's the impact on the distance that we estimate among Drosophila genome skims. How much error do we have? So these are skimmer distances. Before we do any Kraken filtering, we can have high error, like 20% error in our distances if we have 25% contamination. There we have two lines. Then we do like removal of contamination. When the contamination is close to something in your reference database, Kraken does a very good job of removing those contamination, and we get the right distance. Yes, this is all this. This was all for Kraken 2. So I should have mentioned this is all Kraken 2, not old Kraken. Now, when the stuff that we are searching are more distance, when our contaminant is more distant from the reference library, if it's 5 to 10%, Kraken still reduces the error. You go from this much error to this much. It's less than 10% error in the estimated distance. So that's actually not super bad, but hopefully we can do better. And when it's 15% or higher, it's not good, right? We can't improve that level of error much. Okay, so that's where we are with Kraken. Now let's see if we can do Kraken 2. Let's see if we can do any better. Can we increase the sensitivity in 5 to 15% range? And this is important because reference libraries are going to remain incomplete for things like bacteria that are very diverse. what we are going to have to do is allow inexact k-mer matches. Kraken also does enable inexact k-mer matches through a mechanism called masking. It like for every 31 mer, it masks 7 positions in it. But we're going to do something slightly different. So what we are going to try to do is answer the following question. For every read, does it have enough k-mers? You get to decide what we mean by enough. For example, 1 k-mer that has a Hamming distance that is smaller than a certain threshold to some k-mer in your reference database. By default, we are going to go for a Hamming distance of 3. So a k-mer length is 32 by default. So we want to find a Hamming distance of 3 out of 32, so 10%. So a read of 150 base pair, does it have any k-mers that is 10% distant to something in the reference library? We want to answer that question. So if it is, we're going to call it a contaminant, basically. So the way it would work conceptually is you have a lot of reference genomes, like tens of thousands or hundreds of thousands of reference genomes. You're going to get all their k-mers. You put them in the reference library. And for each query, you're going to look at all their k-mers, compute their Hamming distance to all these k-mers in the reference library. If the distance is low enough, you're going to call it a match. Now, immediately, you're going to go like, oh, this is not going to work, right? Because how are you going to compute the distance between every k-mer in every query with every k-mer in the reference library? And you would be right. That is impossible to do. But there is a little trick called local sensitivity hashing that you can use. I'm not going to go through the details of what that is. But basically, what it allows us to do is take every k-mer in the read and compute it and compare it with very, very few reads, very few k-mers in the reference library, let's say 200 of them. And if the k-mer in the query matches any of the k-mers in the reference library, it's going to be one of these 200 with very high probability. That's the beauty of local sensitivity hashing. And that's what we are going to use. Now, so that's the good thing with few comparisons, we're going to find the right k-mers. The way we have implemented this, the bad thing is that it's going to require 120 gigabase pairs of memory if you want to keep 8 billion k-mers in memory. Now, why 8 billion k-mers? Because that's kind of like the right level if you want to represent the diversity of bacteria, like the entire diversity of bacterial life. So bacterial and archaeal life, so prokaryotes, right? So if you take like something like GTDB, which is a database of all, like, a representative database of bacterial and archaeal species, you extract all k-mers and you will get something like 20 billion k-mers. You don't need to have every single k-mer to be able to do this, but you can't go like to 1 billion k-mer. 8 billion k-mer is a good compromise. 120 gigabase pairs of memory, like 5 years ago, that was too much. These days, most server, like I don't think you can buy a server anymore with less than this much memory. So it's not a big problem. You can have that much memory. But it's not something you can run on your laptop, unfortunately, at least our current implementation. Maybe we'll fix that in the future. Okay, so this is something about local sensitive hashing. I'm going to skip it. The method that we designed based on this idea is called Consult. It was published like this month, like earlier this month, it got published. I thought it was previous month, but it seems like it was this month. So it's a very new tool. Just like Respect, it's a little bit clunky. It's actually clunkier than Respect. Installing it is actually very easy. It's just a C code. You compile it, it installs. The problem with it is that if there is any error, it has very bad reporting of errors. It doesn't tell you exactly what went wrong. It just like fails on you. So we are going to try to fix that soon. I apologize for that. In the meantime, let me quickly show you some results, and hopefully we get to do a quick demonstration. Okay, so this is comparison to Kraken 2. So remember, this is the same figure we were looking at before. As the distance to the closest match goes up, once you get to 5 to 15%, Kraken didn't have such great recall. We improved the recall, and also we improved the recall for 15 to 25%. That's quite a bit of improvement, and it's going to make a big impact on the distance. And we didn't increase false positive. False positive is essentially the same as Kraken. Now, what happens if I compare to other tools? Kraken is not the only tool. There is Bowtie, there is Clark, there is all sorts of tools out there. Bowtie and Kraken worked pretty much identically for us. Bowtie just was slower but had lower false positive, so that made no difference. If you use Clark in its default setting, is not quite as good. There's something called Clark sensitive. It really increases the recall, but that's not useful for us because in our case, it was also giving us 60% false positive. Basically, it was also removing non-contaminants, like good reads. It was removing good reads as well. So that's not useful, right? So among all the tools with low false positive rates down here, our method consult had the best recall. Okay, so that's just some results. We looked at those Drosophila data sets we were talking about previously, and basically these arrows are basically showing what's the error in the distances computing between two pairs of Drosophilas before filtering, after filtering from each of these genome skims. And by error, we mean like compared to the distance you get from the assemblies. So the ones that are blue are the ones where like the actual error was reduced. In some cases, actually, this filtering increases the error. The reason is when error is low to begin with, if you remove, like there is some chance that there's always like 5% of real reads are being removed because of false positives, and those can increase the error. But we don't have, there's only one case where after filtering you have somewhat high error. Even this is less than 10% error. But before filtering, you could underestimate distances by as much as like 40%, and that is helped a lot. There are a couple of cases where we were not able to remove enough contact. So in this database, we are not spiking in contamination. This is like real contamination. We are only removing bacterial contamination. My guess is that these are cases where we have non-bacterial contamination where our filtering didn't help us much. So this could be, for example, the kind of thing that Andre is talking about, cross-contamination between samples. We don't have a good solution for cross-contamination between samples. So if these samples are all of the same like kind of a species, I don't yet know how to solve that problem. That's a difficult problem. Okay, so yeah, so for this real genome skims from Drosophilas, accuracy improves. Let me just quickly tell you who did what, and then we'll do a little bit more tutorials. I forgot this yesterday. I don't want to forget it again. So Nora did all the contamination stuff, consult. Metin did the MISA, so the mixture analysis, and Shahab is the one who did the respect paper, genome, the repeated spectra. All of these works are collaboration with Bafna. My colleague here is UCSD. And a lot of these ideas have come about from our discussions with Tom and Christine. up in Denmark, not too far away from where you guys are. Okay, let me take a breath and go back to the tutorials. I'm gonna give everyone just 2 minutes. I think you have heard enough of my voice. You're probably tired. I'm gonna give everyone 2 minutes of respite, and then you have to listen to me again. Okay, so let's start the discussion. So I'm gonna go back. So let's first, remember we started run under Phylogenetic genomes while we were talking, it finished. It took, how long to finish? Yeah, it took also like about 5 to 6 minutes to finish. On the 4 Drosophila genome skims that I have here. And I'm like just looking at the respect output again. So these Drosophila genomes are about half X coverage. I think we did like a fixed size. The genome lengths are estimated. Unfortunately, I forgot to add like assemblies in this case, so we don't know what the truth is. But at least what you can see is that in some cases the repetitiveness can be a lot less. In these are not simulated genome skims. These are from a real Illumina runs. So each of them has a different like error rate. My guess is that these are like all done. Like every run of Illumina have somewhat different error rate, and you can see that 2 of them have very low error rate. Sorry, one of them has super low and a little bit higher error rate, and another one is in between. Uniqueness again is not the same, and high repeat coverage. You see these numbers are much bigger now. Like there are 120 highly repetitive k-mers per million base pair in this case. Okay, so that's just like the respect run that finished. Now, I did something sneaky while we were resting. So let me just tell you what I did. Because consult requires 120 gigabytes of memory, I created a new interactive like session for myself. My old one had expired anyway. So what I did was I increased the number of threads to 8, and I gave each thread 16 base pairs. Sorry, not base pairs, gigabytes. So if you just multiply that by that, you get 128. So that's enough for consult. Consult requires 120 gigabytes, and 128 is going to be plenty enough. So this interactive session is now going to let me run consult. So let's run consult. So I am going to go back here and create a directory for consult runs. So this Drosophila genome skims that we just ran a skimmer on, I'm going to also make a link to them. These data, you should have access to them. It's part of this teachers directory that you should have access to. So let's look at what's inside Drosophila. We just have 4 FASTQ files, right? These are the ones that we just analyze here with respect. So now we want to run consult on them. So let's do that. Before I run consult, so this is going to be the tricky part when you guys try to run consult. We need a reference database. And just like we need 120 gigabyte base pairs, why do we need 120 gigabase pairs of memory? Because we are going to load this entire reference library into the memory. So let's do something quick. I have already downloaded this reference library here on my account. You should have access to it. But you can see it's 120 gigabase pairs. If you want to download it somewhere else, it's going to take a little bit of time, unfortunately, to do that. And there's no way around it. This can't be zipped. Like the way it is already condensed. you can't quite condense it any further. So yeah, it's going to take that long to download. But anyway, so what I'm going to do now is I'm going to create a link to it. Never copy something that big, just create a link to it in my current directory so that I have the reference. So this is my reference. It's built based on GTDB. GTDB, when we looked at it, it has like, I think 30,000 bacterial genomes or something like that. Bacterial and archaeal, both of them combined. And this is going to be our Drosophila genome schemes are going to be our searches. So let's start the search. It's going to take a minute to run. And in the meantime, we are going to do a bunch of cool stuff. So when you run consult, it tells you, it starts reading that reference library. It's still reading this 120 gigabytes. It's reading it. It's going to take some time. Oh, I gave it 2 threads. Let me stop this. This is going to, oh. Okay, what happened? I don't know what happened. I doubt it actually ran. Okay, let me try to give it more threads. This -t is the number of threads. You want to give it at least like I have 8 threads here, so I'm going to give it all. Oh, maybe I didn't get enough memory. So let's see. Maybe I'm not. Oh, maybe. Okay, so it failed last time. I don't. So you can do top to see what's going on. Let me do top U so that failed again. It wasn't failing earlier today. Let me see if it's a memory issue. Top US Mirarab. Okay. So you see, this is taking more and more memory. I think it's gonna start. Oh, it failed. It fails as soon as it gets to 30 megabytes. So maybe I didn't ask for enough memory. Let me try this one more time. So it's more in the. I think somehow I'm not. Oh, sorry, I forgot to ask. Okay, so sorry, I have to change that script. Let's see. There is another command that this. I think it's called number of CPUs per task. Does anyone know this? So I think I need to go like number of tasks is 1. Then there is another thing for number of CPUs per task. I have to give it that one. Let's see if I can find out what that is. 1 second, I made a mistake here.
There's minus CPUs minus per minus task. Is that the one that you're looking for?
That is what I'm using. So minus CPU per task.
Yeah, and then you put equals and then.
Okay, there you go. Okay, this should work. This is different from my, a little bit different from the clusters I'm used to. So let's hope it works this time. Note that I haven't even like activated the Conda consult is not part of Conda. I'll tell you about installation in a second. Let's just see if this is can't open file. Oh, I'm in the wrong directory. Okay. Let's go back to the right directory, which was user work, consult. Yes, this is the right directory. Okay, let's try to run it one more time and hope it works. So while it's running, I'm gonna monitor the CPU. Sorry, the memory. Okay, yeah, this time is working fine. See, it's going up. So now it's got 50 gigs, 60 gigs. It's just reading the library right now. It's not doing anything fancy yet. ID 90 is almost done. Okay, so I'm gonna go out to see what happens next. It's gonna tell you, okay, I'm done reading the library. I'm gonna start doing this search. So let's look at the log file. Maybe I can tell you what some of these things mean in the log file. -C means how many k-mers in a read need to match the library before we call it a match. Default is 1. You can change that if you want. 8 threads. So this is how many k-mers we have in this reference library. If you parse the number of zeros here, it's 8 billion. So guys, I know I'm going a little bit late. Give me another 5 minutes. This should be done. Then it's basically just saying, like, these are all. You can ignore all this. It's basically all the internal parameters, at this point so it just took it 28 seconds to read that library. Now it's doing the actual work. We can even monitor that. So let's go ahead and monitor that. So as it's running, what it does is, we gave it 8 threads, so it has plenty enough. So it's working on all 4 genome skims that we have at the same time. And basically it's going through one read at a time. And if they match one of the bacteria, so if it matches the bacterial database, it's going to remove them. And if not, it's going to add them to this file, unclassified. And then, or files already were searched against human ones. So that's why they had this unclassified underscore to begin with. I didn't do the human removal step here. That was already done. So, you can see that, it's running and it's kind of like adding more files to adding more lines to these output files. It's going to finish in a couple of minutes. So hopefully we won't be over time too much, no more than 5 minutes. And we will look at the results and we'll call it a day. Any questions while this is running? Any questions about consult or any other part? Sorry, everyone. No, we are going over time, but we did hit a couple of snags that I hadn't anticipated. What about cleaning as assemblies? Is it feasible? Yeah, so in principle, you can take an assembly so I mean, that's a tricky question. You guys ask very good and tricky questions. So if what you mean by cleaning assemblies is take an assembly and see if there are any parts of it that match bacterial genomes very well, yes, we can do that with this methodology. We can kind of chop off the assembly into windows of length, let's say 200 or something, or whatever length you're interested in, and just run that through consult. And it will tell you which windows match bacterial database very well. And if there are several windows in a row that match bacterial database very well, you can go ahead and call that a contaminants. But I mean, of course, there are also cases where that is real. I mean, there are ways in which there are longest stretches, but by long I mean like maybe, I don't know, a couple of hundred base pairs, where you have like real similarities between bacteria and something else for whatever reason. Yeah, there are real biological processes that can generate that, and then you would like incorrectly detect contamination. So for example, it could be like a retrovirus that is very similar to a phage, and it's got inserted in some bacteria, and it also got inserted in yourself, like in your genome assembly, and you would think that's contamination, where it's like really just retroviruses, things like that, right? Yeah, but that was a very good question. So the tool finished. Let's look at the log. It tells you how long it took. It took 5 minutes in this example. So not too bad. And basically it produced these outputs, which if we compare with our inputs in terms of like how many lines there are in them, the difference between those would be what's removed, right, as bacterial. So for example, like this first one, the difference between these two, so very like a small portion was removed, but some stuff were removed, and the same is true for this second line, right? So these are now free of bacterial contamination, and we could go ahead, for example, and run respect on it. If we weren't out of time, I would run respect on these, and I would compare the results from the respect after contamination removal to before, and you would, I think, see changes in genome length and repetitiveness and things like that. But we are out of time, so I'm going to skip that step. In the output, it tells you for each of the genomes how many of the reads were not contaminated, how many of them were contaminated. So these were not like super highly contaminated. Okay, so I'm done with the tutorial. I'm going to just take the questions. So there was some questions about blob tools and blob plot and species assignment. So consult right now, as constructed right now, doesn't give you species assignment. It just tells you whether it matches this database. So it's very like high level assignment. I would say like domain level or family, like if you're talking about Drosophila or talking about insects, it would be like order level. For bacteria, it's kind of like domain level. It's very high level assignment. You can build libraries of existing genomes at very high level. It doesn't distinguish the species within, well.
That's the same for blob tools. Like the whole point is to be able to visualize on like the coverage of individual scaffolds and their GC content together with the like a higher rank taxonomical assignment. And if you have like a blobs of scaffolds that are somewhere with a deviated coverage GC content and assignment, is a very strong signature of.
Oh, okay, that makes sense. That makes sense. I didn't know what blob tool was. Okay, that makes a lot of sense. To how many samples does Apple's scales? Apple's is ridiculously scalable. You can, so not MISA. MISA is slow, but Apple's, it just basically takes no time. You can run it a very large data set, a large number of samples, it would be very fast. A skimmer is a little bit slower. You have like the step that is for computing distances. If you have 100 low coverage samples and 100 high coverage samples, you don't need to come, if you are just placing them on a tree with, I don't know, let's say 20 genomes, you should be able to do that. I mean, it depends on how many threads you have. If you have 200 threads, it's like half an hour. If you have 20 threads, it's, I don't know, 5 hours, something like that. Would you do it iteratively? You can do it iteratively, but you don't have to. You can create a folder. Say, maybe I should have shown this in my tutorial. You can create a folder where you have all of your queries, just like I did right here for Drosophila, right? And you can compare the distance between these, all of these queries and all of your samples in one skimmer run. So you run it once, it computes all of them at the same time, and then you run Apples once, it's going to place all of them at the same time. So no, you don't have to do it iteratively. You can do it all in one run. All of these tools are multi-threaded. So if you have multiple samples, they're going to use different threads for different samples and save you some time that way. Other questions? Any other questions, comments? Thank you all. This was very fun to teach because you all asked very good questions, and that makes it a lot more fun for me.